Hello everyone, my name is Julianne. I am a medical oncologist originally from Brazil, currently working and living in London as a clinical research fellow and co-investigator of the Royal Marsden Hospital. I was a PPCR student in 2019 and ever since I've been fortunate to be part of the team as a teaching assistant. This year, I'm applying for senior teaching assistant post, which I believe it's a huge challenge, is a huge commitment, but definitely worthwhile. And I hope I can be beneficial to the students once again and learn again from them and from this amazing team. Thank you very much indeed, PPCR coordinators, for supporting us towards this difficult time during the pandemic. Today, we're going to talk about basic concepts of clinical research. This is part of a lecture I've given a couple of years ago, and I decided to share here all my gratitude for what I've learned from PPCR. Ultimately, the results of a clinical trial should be able to tell you that you can act upon something to make it behave differently. But what's going to make you a researcher is being capable of explaining what you've done under measurable conditions and how someone could replicate the same findings and obtain the same results. So the slide we are seeing now was presented in one of the most important oncology conferences a couple of years ago, 1 decade ago, with this significant, huge improvement in progression-free survival outcomes for patients with metastatic hormonal receptor positive breast cancer, HER2 negative. This was a combination of treatments trying to overcome endocrine resistance. Basically, the lovely shapes of the curves are absolutely clear in terms of the beneficial effect of the intervention, which is the combination of the two agents, everolimus plus exemestane. And by the time the publication was released in 2012 at the New England Journal of Medicine, I remember the debate was about the hazard ratio of 0.36 and never previously reported for this population. Everyone was very excited. However, unfortunately, although the progression-free survival benefit of more than 6 months was evident on the publication, proof of overall survival gain could not be robust enough to make this standard of care for a long time. Today, the treatment of these patients has changed completely for those who are interested in breast cancer. And basically, I want to give you the example that it's not always when you can measure your variables that you can replicate the same amount of benefit. And in this sense, it's extremely important to understand what we need to prove and how we need to prove in terms of research question, intervention, and outcome. So the beauty seen with some results makes us wonder, are the results absolutely replicable? Well, they may well be, but not necessarily as you wished. If that's going to be clinically meaningful, that's going to depend on your target population and obviously the context in which the background knowledge is going to be applied ultimately. The lecture today is going to be divided into different times. I tried to put the topics chronologically divided in before trial, which is basically the phase in which we have the development of the question, the development of the study design, randomization, selection of a study population, and there we have the very important discussion about internal and external validity, which is going to make our study powerful and replicable. And subsequently, we will have a glance of the after-trial processes, including analyzing data, the types of tests to be applied, depending on the type of information you collected, and we will also have one example of what we can do in terms of survival analysis. Next time when we speak again, I hope to give you more information and to have the opportunity of discussing about survival analysis calculation, which is another amazing lecture from PPCR. It's well known to everyone that the development of a research question is perhaps the most important step, because if you have a strong research question, you can produce important results regardless of the positivity or negative correlation of variables. Here we have the acronym, the famous PICOT acronym for the development of the research question, in which you must define from the beginning the population you are going to study, your intervention or independent variable, your control group, or the comparison with the target population, your outcome or independent variable, and the timing in which your experiment is going to be measured. The example one is from a trial called Valley 2. So this is the title of the paper published. Buparlisib plus fulvestrant versus placebo plus fulvestrant in postmenopausal hormonal receptor positive, HER2 negative advanced breast cancer patients. This was a randomized, double-blind, placebo-controlled phase 3 trial. Obviously, to me, as a breast cancer specialist, it's easy to define the different variables, population and timing, but depending on everyone else's expertise, you can easily find the research question described within the article. So here I have written the research question of this paper as: amongst postmenopausal hormonal receptor positive, HER2 negative advanced breast cancer patients, can the combination of buparlisib plus fulvestrant compared to placebo plus fulvestrant improve progression-free survival in the range of time between September 7th, 2012 and September 10th, 2014? So that's the research question. The population is postmenopausal hormonal receptor positive, HER2 negative advanced breast cancer patients. The intervention is the combination of fulvestrant, standard of care endocrine therapy, plus buparlisib, compared to control, which is an active control because patients basically receive standard of care fulvestrant plus placebo, because this is a double-blind study. And the main outcome is the main measurement of efficacy is going to be progression-free survival improvement in the range of time they defined to follow the patients towards 2 years, from 12 to 14. And herein again, we see another example, alpelisib for PIK3CA mutated, which is a biomarker of breast cancer patients, hormone receptor positive advanced breast cancer. The research question of this paper was: amongst patients with PIK3CA mutated, hormonal receptor positive, HER2 negative advanced breast cancer previously exposed to endocrine treatment, can the alpha specific PIK3CA inhibitor alpelisib plus fulvestrant compared to placebo plus fulvestrant improve progression-free survival in the range of time between July 26, 2015 and July 21st, 2017? So it's quite a complex model of research question. However, you can identify, using the acronym PICOT, every item of interest to continue the critical thinking of the research, and that's the very basic first step you must take before writing a grant, for example. So taking the same example discussed before, we're going to work with the development of hypotheses. So the null hypothesis of a research is always the nature. It's always stating that there's no difference between the two groups. And the alternative hypothesis is what you are going to prove, that there is a difference, and ultimately you can interfere with the difference and you can measure and replicate it. So herein I've written what ideally everyone would like to prove with the alternative hypothesis of trials. There is a statistically significant difference, I can quantify it, and I can tell you that if we repeat this trial 100 times, the result will be positive in 97.5 over 100 times. In this particular trial, they assumed a 2.5% type 1 error at random and a 5% type 1 error two-sided corrected by Bonferroni. Very elegant design. The variables in this study, as we described before, the intervention or independent variable must be controlled by the investigators. And why is that? Because in experimental trials, you must prove a relationship between cause and effect. And you can only do that if there is a variable that you can control, and that's going to be your independent variable, because you can manipulate it. On the other hand, the dependent variable cannot be controlled, is what you want to modify with your interventional treatment, for example. In this case, it's going to be survival. Obviously, there's going to be a group of covariables which can interfere with your outcome independently or dependently of your intervention. So it's very important to have the expertise in the field to define which are the important confounders, no, but covariables that must be controlled. We are going to speak about confounders in a couple of slides further. So the intervention in this case is alpelisib plus fulvestrant, and the outcome is progression-free survival. The covariables to be controlled, as you must have preempted already, are clinical features, for example, age, previous treatments, biological behavior of the disease, and biomarkers. And these, if they are so important to the point that they can definitely interfere with the effect of your intervention, it's important to control as stratification factors or subgroup analysis. But it's important to ask beforehand what's going to be the cost of a stratification analysis, of a stratified randomization. Why is that? Because that's going to interfere directly in your study population, in your sample size. So, continuing, a study population must be very, very well defined, because ultimately this is going to be your experimental environment. Your study population is going to be the population in which you will prove your hypothesis or no. And this is very nicely described in Professor Freyne's book, when we have this subset of study population that can be studied, for example, the sample. You also have the subset of target population that can be studied, which is going to be your study population in general, not divided in subgroups. And the study population in your study must be representative of your target population and ultimately the general population. In the cases we were discussing before, we were speaking specifically about a certain subtype of breast cancer, but the patients studied there, they must represent somehow the target population, which is the population of patients with breast cancer. And this may sound simple, but if you consider that you are going to give an experimental treatment to someone, it's very important that the person, the subject, fulfills some basic criteria of fitness and health in general, because you don't want to threaten other comorbidities, for example, with your experimental treatment that you are not sure is going to work. So the first discussion we see when we try to extrapolate results from a trial is whether the patients included in the trial, they are really representative of our overall population, because they were so perfect. They have an excellent performance status, they don't have comorbidities, but they had to be like that to be submitted to an experimental treatment. And then the philosophical discussion, what's going to be the most important point, is if your result is strong enough to be proved, or if your treatment is good enough to be the standard of care. And this is basically the last step, the result of a good selection of study population. So what to consider when you are selecting the population to be studied, which is going to be your experimental environment. The phase of the study, 1, 2, 3, or 4, and here we must recall early phase studies, they are trying to prove safety, and late phase studies such as phase 3, they are trying to prove gains in efficacy. Response to treatment, and obviously safety is key. Safety is a paramount. So if you don't start by safety, you cannot proceed, regardless of the phase of the trial. But obviously, the outcomes, sample size, calculations, and extrapolations, they must be aligned with study design and with study population in terms of arrangements. So inclusion and exclusion criteria are extremely important to be fulfilled in order to make sure your study population is correctively and assertively selecting, including the subjects which are more likely to benefit or not from your experimental treatment. And therefore, your results can be extrapolated if they are true. and as we were speaking now, you can preempt, we are talking about internal and external validity. So internal validity is basically the reliability of your results. How accurate are the conclusions? And external validity, as we said, is basically, can the study population be taken as a reliable sample of the target population? And how accurate are the results when extrapolated, when generalized to the target population or the real world population? Obviously, if you want to strengthen your internal validity, you must reduce the variability. Why is that? Because if you include a broad a study population with different variances, with different covariables interfering in your outcome, you're going to lose the power of your benefit. You will miss the opportunity of showing the best result you could achieve if you had the best subject. Obviously, studies with very high internal validity, they have less variability and they have increased power, which is going to be a higher internal validity. On the other hand, you have a decreased potential of generalizability. Which are the pitfalls of these studies? obviously bias and confounders. And what is the difference between covariables, bias, and confounders? You cannot control. So basically, what is called covariable is something that you can somehow predict or control, or by stratification or by post hoc analysis. So bias, herein we have some examples: selection, performance, ascertainment, recall, attrition. These are common types of bias which can reduce the validity of your study, the internal and external validity regardless, as well as confounders. And we'll have a nice example further. So what can prevent you of having to deal with the risk of bias and confounders in your results? Blinding and randomization. So we know there are different strategies of randomization, such as simple randomization, when the computer software is going to give you a number and this random number is going to select whether the patient is going to be allocated to one or another arm. That can be problematic for smaller populations, as you can, as there could be imbalances between the arms. Block randomization, where blocks, for example, predefined blocks of 4 patients will receive 50% interventional treatment and 50% control group. The problem with block randomization is obviously if the investigator is not blinded and he knows that the blocks have each block has 4 patients and 2 patients were already allocated to the control, he's going to know that the next 2 patients are going to be allocated to the interventional arm. And therefore, the collection of data, the assessments may be biased because the person will obviously be aware of the treatment allocations, potential side effects, or potential beneficial effects. The solution for that is blinding, or which is not possible sometimes. Obviously, because if the patient is taking something and starts presenting side effects, you're going to know that the patient has got interventional treatment instead of those who are taking placebo and no side effects at all. The solution for that, alongside blinding, is having variable block sizes of randomization, variable between 4 and 6, and the investigator doesn't know whether that block is going to have 4 or 6 patients, so it's impossible to predict what's going to be the next sequence of treatment, even though the first 5, for example, have got control, have been allocated to the control group. It is also possible to stratify groups and randomize after stratification. It's a very, very powerful way of proving something with blocks, because the randomization is going to make sure the distribution, the allocation of treatment is going to be equal amongst different strata of patients. Does that make sense? I hope so. And here I put a very classic example of one of the most powerful studies in oncology. One of these studies with, let me correct myself, in statistical terms, one of these studies in oncology with the highest internal validity, which is the Cleopatra trial, which had 90% of the population with no previous exposure to anti-HER2 therapy, which is one of the most important treatments for this population of breast cancer patients. So 90% was absolutely naive of treatment. And this study showed a great benefit for those patients. So could that, the lack of previous exposure to the treatment, had been a selection bias to include diseases more likely to respond to the interventional drug? And the answer is no. Because there could have been a selection bias if the control group had not had a comparable proportion of patients with this baseline characteristic. So selecting a study population is absolutely different from selection bias. Bias is when the populations are not comparable. If they are equally distributed and the errors were predicted before, Obviously, the randomization, assuming the randomization was adequate and appropriate, the results can be replicable with no risk of making a mistake. At least in 95% of the cases, assuming an uncertainty of 0.5%. Herein is speaking briefly about study design, which is a big topic in interventional trials. Experimental studies try to prove a cause-effect relationship. And the study designs, the most frequent study designs for experimental studies are parallel, where you follow the subjects during a period of time. And here in parallel study, you can study one independent variable or two or more independent variables. However, having more than one independent variable, it's important to quantify the effect of each one towards your outcome. And in this sense, factorial designs are highly recommended. They are very strong because you can answer two questions at the same time. However, the interpretation of the results is difficult. The recruitment is challenging as well because it needs to recruit more patients for different arms. And not very well accepted sometimes. It's important to define at the beginning if you're going to have a placebo control or an active control. As I showed before, patients receiving standard of care, active treatment are compared to patients receiving standard of care plus something else. And there are also 3 arms studies, parallel studies, in which you have obviously one control group, one interventional group, and this needs to be defined beforehand, very well defined, because that's going to define your sample size. But you have also another cohort of patients, perhaps with different independent variables, but your study was not designed to answer the question whether that alternative combination is better or worse, for example, than your control. So parallel studies of 3 arms, they are important for comparison, but the power of your study and the design of your study, sample size calculation and errors are defined only for 2. Well, I'm explaining this very basically. I'm pretty sure during the statistical analysis module, we can discuss this further with more details and much more information than this brief chat we are having here. There are also studies in which you have the same individual and repeated measures, like the crossover design, which is an interesting design when you want to expose one patient to different types of treatment and measure the effect after each treatment. Important to be aware of carryover effect, which is basically the first treatment, for example, still having an effect when the patient receives the second treatment, and therefore the outcome is going to be interfered not only by the second treatment, but the first was still making a sort of effect and could have been a synergistic effect or jeopardizing the effect of the second therapeutic intervention, if that makes sense. The order effect can interfere on your outcome as well, in terms of whether the second treatment, for example, was less effective because the disease was more resistant after being exposed to the first one. And the learning effect. This is more for patients who are, for example, answering questionnaires. If they have to answer the same questionnaire more than once, they're going to learn the questions, and it's more likely that they will complete everything afterwards because they know how to answer. Herein just a couple of examples of a factorial study. The study COGB 40603, they tried to answer whether the impact of carboplatin plus bevacizumab to neoadjuvant protocol of chemotherapy for patients with breast cancer could be beneficial or not. And they had two different questions, only to see whether carboplatin made a difference or whether bevacizumab made a difference. But obviously the combination of both is going to be challenging because the comparison is not going to be accurate. And herein we start wondering about the most challenging analysis from factorial design, which is controlling interactions of variables between the arms. Here we have another example of study. This is a parallel design. Patients, the study population is stratified by the factors you can see. They are allocated between two, they are randomized and allocated between two arms, including standard of care chemotherapy and a standard of care chemotherapy plus immunotherapy. Obviously, oncologists are going to debate with me whether nab-paclitaxel can be considered standard of care chemotherapy, but that's going to be a nice debate for another time. But that's definitely a landmark study for patients with triple-negative breast cancer, in which immunotherapy showed improvement in survival outcomes. So, talking about confounders, and this I've also got from Professor Freyne's book, to understand the importance of the types of tests to be applied on our results after the study is completed. And here we start the phase after trial. A man gets drunk on Monday on whiskey and soda water. He gets drunk on Tuesday on brandy and soda water, and on Wednesday on gin and soda water. What causes his drunkenness? Obviously the common factor, the soda water. So that's funny because we see this quite a while, and probably some of you are now remembering observational studies, but in experimental studies, if you don't control very well your variables, your covariables, and if you don't predict confounders, that can be a huge issue. And tests cannot identify bias or confounders. First of all, before the analysis and before calculating your sample size, we know we must have some assumptions, such as the statistical errors we want to take the risk of beforehand, like type 1 or error or alpha error, rejecting the null hypothesis when it is true, or type 2 error or beta error or power of study, accepting the null hypothesis when it is false. The description, this is a basic summary of types of data and quantitative and categorical data. Basically, the quantitative data represents amounts. The quantitative variables, they represent amounts and can be discrete, such as number of students, number of trials, or continuous variables, such as age, volume, time, distance. And the categorical variables, they represent groupings. Binary, for example, yes or no variables. For example, patients, pregnant patients, if it's just yes or no. Death is just yes or no. But don't confound death with survival. Survival is is represented by days, times, seconds. So it's a continuum. Death is just yes or no. Is alive or no. Nominal variables, species, brands, molecular subtypes, for example, or ordinal variables, for example, scores of pathological response, performance status of patients, and herein I described a very nice way of analyzing retrospective data. For example, when you want to categorize continuous variables to make it easier to interpret and also to accept. For example, weight. It doesn't matter if the patient has lost 1 or 2 kilos, if that's not clinically meaningful. So if you have a score that can assertively say whether the patient has a good state of nutrition or not, or is obese or overweight, such as BMI, that's more clinically meaningful than the weight itself. And here I described another example also for oncologists, clinically significant amount of response categorized by RECIST criteria, which is a very specific way of measuring response in oncology trials. Proportion of patients alive and whatsoever. This is a basic example of how to identify different types of variables in a trial. Again, speaking about IMPassion 130 trial for immunotherapy amongst patients with triple-negative breast cancer. The intervention was atezolizumab plus chemotherapy nab-paclitaxel, which is a categorical variable, and the outcome was survival, which is a continuous variable. These stratification factors were the receipt or not of neoadjuvant or adjuvant taxane, which is the same class of nab-paclitaxel, and that's a binary outcome if the patient received yes or no. Presence or absence of liver metastasis at baseline, yes or no, binary. PD-L1 expression, which is a biomarker, positive or negative, it's a binary variable as well. And the two primary endpoints were continuous. PFS, progression-free survival, in the intention to treat population, PD-L1 positive, the biomarker, and overall survival in the intention to treat population only if the findings in the first population was significant. So that's a very, very special way of gathering two primary endpoints. And this study particularly had an amendment to include more patients. This is an example. This was one of my first bonus assignments. The trial had 90% power for the primary analysis of PFS amongst intention to treat population and 88% of power for the overall survival analysis. That's in oncology, that's interesting. After receiving the data for whatever variable you need to analyze, we will also need to see the distribution of patients, the distribution of that specific data you are collecting, whether that's normally distributed or not, allowing us to apply parametric tests such as t-test, ANOVA, and linear regression. The parametric tests, they need to have the assumptions checked, such as continuous data, if the data is normally distributed or not, and non-parametric tests, such as Kaplan-Meier survival analysis, do not have assumptions. I've given the example of Kaplan-Meier because it's one of the most common tests to be applied in survival analysis and in important trials as well. So it's not, you can obtain important and significant results using non-parametric tests, but it will depend on your population, on your study design, and obviously in the internal validity. It's gonna rely a lot on the concepts of cause and effect of your intervention on your outcome. This is going to be a very nice lecture during the PPCR course when we will be able to see the professors and Professor Freni describing how we can apply different tests, parametric and non-parametric, comparing groups, basically depending on the types of variables. For example, comparing two groups with independent variable binary. If the distribution is normal, you can use the parametric unpaired or paired t-test. However, if the data is non-normally distributed, you must use Mann-Whitney-Wilcoxon. And for the analysis of categorical data, you definitely can have a very good analysis from chi-square, or if that's binary, is still, you can apply other tests as well. To compare 3 or more groups with independent variable, categorical independent variables, you can, if the data is normally distributed, it's possible to apply ANOVA analysis, or if it is non-normal, for example, when you want to just describe potential interaction between side effects, which are categorical data, normally, non-normally distributed, you can use Cruz-Coalis, and the associations can be made by chi-square or Fishers. Associations between 2 variables, if they are continuous, there is the Pearson correlation and Spearman correlation. Association between 3 or more variables if they are continuous, and then you start using, if they are normally distributed, multiple linear regression, and if you have categorical data included in your analysis, you may want to use multiple logistic regression, and that's the ultimate stage of the statistical module of the course. Obviously, we will have opportunities of seeing brilliant lectures about this during the course. I've given here example of how to interpret types of data after having the information from the trial, the same trial we discussed before in Passion 103 and 30. This was a positive result. I'm sorry because it's a busy slide. I'm gonna pass to the next one, which is more relevant. I'm basically describing why did they choose the tests they did to perform the statistical analysis of this trial. The methods of regression were used because the distribution must be sensory independent. Obviously, patients, they die, they lose follow-up for different reasons, and it is important to control for confounders as well. So this is the beauty of using regression models as well. PFS and overall survival were compared between the arms with the use of a stratified log rank test, which is a non-parametric test applied for skewed distributions or for data censored, assuming that censoring will not interfere in the final measurement of the treatment effect. The hazard ratio for disease progression and death were estimated with the use of a stratified Cox proportional hazard model, which is a regression model of survival analysis as well that works better for categorical variables when analyzed as predictors, which is the case. For example, if the patient is PD-L1 positive, if she would have a better response or not. So they would like to be able to predict whether this could be I wouldn't say a surrogate marker of response, but definitely a prognostic biomarker, for example. Kaplan-Meier analysis was applied to progression-free survival and overall survival, which is also appropriate for survival analysis after passing specified time points, because it's a time to event analysis, and accomplishes the fundamental assumption of sensory independence data. The comparison of response rates between groups was made with the Cochrane mental test, which is appropriate in this setting for comparing 2 variables controlling for important confounders. So the take-home message of basic concepts of clinical research in this very brief talk we had today are: a strong research question should produce relevant knowledge, regardless of the results. The appropriate study design will answer the question, and negative results can be hypothesis generators as well if the question is strong. Positive results may never be replicable. P is not a surrogate of external validity. Is only a degree of uncertainty. Be aware of uncertainty and work with margins of errors. before you go and replicate your results, wait for the publication of the paper. So again, obviously, art is more difficult than clinical research. As you said from the very beginning, this is one of my favorite masterpieces, which inspired me to have a brief thought. Replicable results are standard. Outstanding findings are unlikely to be repeated if the time has passed, and miracles are miracles. And I'm pretty sure everyone knows what's standard and what is a miracle and what we do. I hope this lecture has been somehow useful and enjoyable. And again, thank you very much for the opportunity.